I'm JONG MYONG Kim in Riken Institute, Plant Science Center. So we focused on just histone deacetylase 6, HDA6 in Arabidopsis. So basically, HDA6 regulates gene activity by removing the histone acetylation from the activated site and to make inactivate and silent chromatin. And HDA6 is concerning several biological processes in Arabidopsis. For example, silencing of the genomic elements like repetitive sequence depletion and the transgene silencing, and also like them. And also gene regulation of biological processes. Recently, we reported about cold acclimation, HDA6 function in the cold acclimation, and also jasmonate and ethylene signaling. So actually, there are many reports their article was reported, but direct target gene of the HDA6, the histone modification site, functional mechanism, and genome-wide function of HDA6 had not been determined. So recently, we reported about the mechanism of the functional mechanism of the gene silencing by HDA6. This is just image. The model is like that. On the HDA6 direct target gene region, DNA mesylase MET1 and HDA6 cooperatively function to the repression of the gene silencing. This is the first step of the gene silencing, and then after then, CMT3 and some histone mesylase will come into this region. So that HDA6 and MET1 cooperatively function in the genome region to replace from active mode to repressive mode. So in this meeting, I especially focused on epigenetic regulation for transposable element silencing by HDA6 on genome-wide. At first, we established the chip-chip and also chip sequence system in our lab. This is a part of the result of chip-chip and chip sequence data analysis. Upper two columns are chip-chip data, and lower two columns are chip sequence data. You can see the very good peak in this panel. So, and the resolution is very good. And the correlation of those data are also highly correlated. And also, you can see this region. So in HDA6 mutant, histone H4 hypersaturation is highly accumulated, but in wild type, is no accumulation. There are no accumulation. So that is on the gypsy-like transposable element region. We try to make a profile the HDA6 binding using a specific antibody we generated, and also histone H4 hypersaturation mode. So we determined about 10,000 peaks of the HDA6 binding peaks. So these peaks are distributed on the about 1,500 gene region, and also on the 400 transposable element gene region, transposable element region. So this is a Venn diagram of these HDA6 and hypersaturation data. Today, I will talk about focus on the transposable elements. This merged region, so just 150 transposable elements are positively regulated, directly bind and affected by HDA6. And so this region, about 1,000 transposable element genes are just changed hypersaturation. So that we think this region is indirect target of the HDA6. And also, we also find out the just binding region. So this is one of the potential target of the HDA6. To assign the genome-wide binding of the HDA6, so we try to plot the HDA6 binding location. And when I set the very high threshold, and then we can see the you can see that HDA6 binding is highly accumulated in the pericentromic region. So this is just an example, just one example. So on the chromosome 3, so but actually all chromosome on the all chromosome HDA6 highly accumulated on the pericentromic region. And to resolve the HDA6 binding and enrichment of the H4 hypersaturation accumulation, so we try to find out which transposable element families are derived by HDA6. So this graph is the distribution of the hypersaturation on the AGI-coded transposable element region. The blue is in wild type, and red is in HDA6 mutant. So actually, the hypersaturation enrichment of the hypersaturation is increased and shifted in HDA6 mutant. And statistically, we determine which transposable family is needed to such a high accumulation of the hypersaturation. So our result, so line family and also gypsy families are specifically hypersaturated. So that this shift is derived from those two specific families' hypersaturation. So, and also we try to mapping such a hypersaturated gypsy and line location on the genome-wide. So surprisingly, hypersaturated gypsy and gypsies are located on the pericentromic region, more near center. And in contrast, hypersaturated lines are located on more, just a little bit difficult to see, more outside of the gypsy located region. So from this result, it suggests that the pericentromic regions are structurally relaxed in HDA6 mutant. And actually, we also tried to identify the K9 dimethylation and also K27 trimethylation distribution on this using this mutant. From our data, K9 dimethylation is correlated to this phenomenon, but K27 trimethylation is not. So, and also, moreover, we found very interesting data, the phenomena. So, in HDA6 mutant, the very specific target is overexpressed and also maybe jumped. We checked copy number variation and gene expression of the specific copier transposable element. In HDA6 mutant, the copier transposable element targeted by HDA6 were highly expressed, and the copy number variation numbers are increased in HDA6 mutant. This graph shows x-axis is the expression, and y-axis shows a copy number variation. Actually, copier transposable elements increase the expression and also copy number variation. This is just image. That is not real. But we try to simulate the location information using very simple program. And actually, the H4 hyperacetylation low size are accumulated on the pericentromic region, and just a little bit relaxed the chromocenter. So this is just image, so that we are now trying to confirm the real imaging data using a microscopic imaging analysis using in situ hybridization. So the conclusion is HDA6 is required genome maintenance via transposable element silencing. All of informatical analysis were done by Dr. Endo in Riken Institute, and also such a simulation of the imaging done by Cycloid Inc. And also this project was done by all lab members in Riken Dr. 6 lab. Thank you.
Wondering, did you say maybe which antibody were you using for the H4 hyperacetylation? And does that recognize all the specific residues which would be
yes, so
acetylated?
that is a commercial one. And that can recognize K5, K8, K12, and also K16.
Mm-hmm. So you haven't yet tried to use any more specific ones to see which particular residues are
For example, I tried K9 acetylation. Yeah, and some are correlated, and on some regions are not correlated. But almost all of the regions are correlated.
So HDA6, I think, came out of Marjorie Matsky's screen, one of her RNA-directed DNA methylation screens. Are you seeing a correlation with certain siRNAs at the loci that where you see HDA6 targeting?
About correlation with the small RNA. So actually, we tried, and we extracted the data from the database and mapped such a data on this genome map. So, but almost all of such a mapping regions are similarly at in how can I say same region mapped with highly acetylated region. So, but we also tried to detect the small RNA in HD6 mutant, the changing of the small RNA in HD6 mutant, but that was very difficult. So in now we can't detect about that. So that I don't say about the correlation is or not.